Hi everyone, welcome to enzymology classes. Today we'll be discussing about one important enzyme, glutathione reductase. So glutathione reductase belongs to the oxidoreductase family of enzymes as indicated by its enzyme number. So as this first digit indicates, it belongs to the oxidoreductase family of enzymes. So the function of this enzyme is to maintain the oxidative balance inside the cell. So the enzyme catalyzes the reduction of glutathione. So this molecule, which is the structure is given here. So this molecule is termed as glutathione. So glutathione has, this is the oxidized form of glutathione. So this oxidized form is joined by one disulfide bridge. So it has two reduced glutathiones joined by disulfide bridge, and when it is acted upon by this enzyme, glutathione reductase, in presence of NADPH, which is a electron donor, this produces two molecules of reduced glutathione, so which is designated as GSH. So by reducing these, it accepts two protons from the cytosol, and the protons are released from various oxidative stress molecules such as hydroxylate ions, superoxide ions, and hydrogen peroxide. So these molecules are also termed as oxidative stress molecules, which are generated through various mechanisms such as the byproduct of oxidation, as well as action of various toxic molecules inside the cell, and these molecules need to be removed. And this protein, glutathione disulfide reductase, or in short glutathione reductase, which is designated as GR, is conserved in all animals, yeast and bacteria. So there are two isoforms found in plants, but ironically, this enzyme is absent from Drosophila and Trypanosome. So Drosophila and Trypanosome has other systems of other homologous enzymes termed as trypanothionine or thioreductase. So these are similar molecules which take over the functions of glutathione reductase in these cells. So they help to scavenge the oxidative stress molecules. They help in clearance of xenobiotics, which are also other forms of oxidative stress molecules. So they maintain the ratio of reduced glutathione to oxidized glutathione at a high level, and they work in concert with glutathione peroxidase, a similar enzyme which is also involved in maintenance of oxidative stress inside the cell. So both glutathione reductase and glutathione peroxidase, they work together to maintain oxidative balance of the cytoplasm. So to briefly look at the structure of this protein, the glutathione reductase from human erythrocyte is a homodimer, and each monomer is 52 kilodalton, and each monomer contains 3 domains. So here we can see together in structure that it has a NADPH binding domain, so this one, and one FAD binding domain, this one, and one dimerization domain. So that is also called interface domain. Interface domain. So this domain is responsible for catalysis. So the overall structure is given here. So there are two amino acids, cysteine 58 and cysteine 63, which are present at the active site, and the overall reaction is the transfer of two electrons in the form of hydride ion from NADPH to FAD, and from FAD to these cysteine residues, and from these cysteine residues to the oxidized glutathione. So this is the oxidized glutathione, which is the substrate, and this converts the molecule to, the enzyme, the cysteines 58 and 63 are reduced, oxidized, which in turn reduce the oxidized glutathione. So the enzyme is oxidized in the first step, and the glutathione is reduced, and upon acceptance of another NADPH molecule, the enzyme gets reduced and NADPH becomes, NADP plus, which was first converted from NADPH, is replaced by NADPH to break this disulfide, and the enzyme regains its original state configuration. So this enzyme is very important for the survival of animals and plants. So it is a thermostable enzyme which is stable up to 65 degrees Celsius. So very few enzymes are there which are stable at this temperature. One such example is alkaline phosphatase. So it is inhibited by sodium sulfite and some flavonoids. So flavonoids are found in plant. So some of the flavonoids inhibit this enzyme, and if this enzyme becomes non-functional or in its absence, so there is a condition called favism and cataract. So we'll be discussing about the mechanism of action of this enzyme in the next video.